All right, class. So today is the 31st of August, and we're going to be go over the material using these lecture videos. Today's topic is the scientific method, and it is section 1.1 in your notes. And because this is the first day of notes, you can decide whether you want to conduct your notes online or in paper. And I will be collecting notes in a Canvas assignment online, but you can submit, photos of your paper notes if you prefer to do it that way. Here's an example of the linked notes. Change the titles obviously to reflect biology, and the first unit is molecular biology. And the topic for today, is the scientific method. You want to keep all of the notes for Unit 1 within this page, so if you need to make a copy of the chart and paste it on for the next page, that's fine. Delete these sections after you read them, and the main body of the notes is going to contain information from the slides, valuable. It could be vocabulary terms and definitions, dates, important characters. And then this column on the left is for questions, thoughts, experiences, other things that you want to put in the notes that relate the content to your own personal experience. And specifically, questions are really important. It's important that you come up with a few questions for every day of notes, because I may ask to have you share those questions. As always, record your attendance before class. Every student will be required to record that attendance. We'll all be using the same Google Meet link, but I've separated students into 3 sections. And I just sent an email describing which students are in either section 1, section 2, or section 3, and what time you will be expected to be present at the meeting. If you're not in the right section, I may ask you to come back at a later time. For today's assignments, the 2 discussion board posts, there's a standards presentation that will be conducted during the Google Meet, and the Penny Lab, all of which I will cover during the Google meeting. So molecular biology is our first unit. If you look at the units for the whole class, you can see it covers some basic biological concepts, and this is going to be the units that you have to look forward to for the whole class of biology. So the driving question today: how can I use the scientific method for everyday inquiry? And you may be familiar with the scientific method. There are specific steps that are involved. First, asking a question. What's the purpose of the experiment? Then forming a hypothesis. This is not an I think or I believe statement. It is also not an educated guess. It has to be testable. So when you conduct a controlled experiment, you have manipulated variables and responding or dependent variables. You only conduct an experiment with one variable in mind that you are changing. Everything else must be the same. The same time, the same temperature, humidity, pH. You want to keep things the same, so you're testing only one variable at a time. And then the control is an item that's used for comparison. This is where you will not change anything. You just leave it and observe the results for that experiment. So the next steps: record and analyze results, collecting data. Things that are easy to collect would be time, weight, any other numerical measurements. And then finally, preparing a conclusion, summarizing the data in a written document or a presentation, and then publishing, allowing others to review and reproduce the work. So the Abiogenesis Debate in 1668, Francisco Redi disproved spontaneous generation, and he was kind of the father of experimental biology. By conducting a controlled experiment and publishing his work, this kind of led to a lot of science, the explosion of thought and recorded science. But the theory at the time was that life arose from non-living matter. If you have nothing, life will come from that. That's what the thought was at the time. And the reason they thought this was if you left meat out, and obviously at the time they didn't have refrigerators, flies would attack the meat, and you would get maggots that would start to crawl out of it. It's kind of a gross thought, but that's just what happens. See, he set up this controlled experiment where he said, I don't think that life comes from nothing. I think that these maggots are coming from the flies. So he set up 2 jars, one that was sealed, one that was not, and the responding variable, the control, is what happens already in the environment. We know that if I leave meat out on the counter, it's going to get flies, it's going to get maggots. But the thing that he's testing is if I cover it with this lid, the dependent variable then would be the flies that arise from that. So the independent variable is the lid. The dependent variable is the response. Are there flies or are there no flies? So Louis Pasteur did another famous experiment where if you heat up gravy and you leave that out over time, it gets moldy. Well, if he first did an experiment where he heated up gravy and sealed the glass container, and no mold came out of it. He recorded those results, and then scientists came back and said, well, everyone knows living things, they need to breathe, they need to have air. So he developed this S-shaped flask, and he boiled up the gravy, no microorganisms. And that was because the microorganisms from the air could not crawl up the S-shaped flask. And even though it was exposed to air, there was no mold that formed, nothing, no bacteria was able to take over and colonize that gravy. So he published his results, and you might recognize his name, Pasteur, because they kind of developed pasteurization, which led to a lot of really advancements in saving food, very important for modern food availability. So again, independent variable is the change in the glass, and the dependent variable is the response. Was there mold? Was there no mold? So that brings us to the end of lecture 1.1. And make sure you record those notes, and we're going to be talking about in detail more of the assignments, and we'll even be presenting our course presentation, or our standards presentations, which are very, very short. For today's penny lab, you'll need a penny and some water and some soap and a paper towel. I look forward to seeing you all in class. I'll talk to you soon.